My name is Alex. I work in the crop protection, ecology, and perennial oilseeds program at the Plant Institute. I'm going to take you around some of my activities today. The plant I work with primarily at the Land Institute is Silphium and Tegrafolium. Today I'm taking some floral traits on these plants. There's a lot of interesting differences. Some plants have larger heads with thicker ligules, and some have smaller heads with fewer ligules. Each of these is a ligule. It really excites me to see all this plant diversity because that's really the foundation of the breeding program that lets us make decisions about which of these traits we'd like to see or would be beneficial to have in a perennial oilseed crop. So today I'm going to use these set of calipers to measure the head diameter and count the number of ligules and measure the ligule length. These floral traits I'm talking about are correlated with seed yield, which is important for silphium as a perennial oilseed crop. Though we're also very interested in traits that are indicators of or related to disease and pest resistance. We have some bees that we're keeping in the greenhouse right now to help pollinate the different silphium plants. Here's a cute sundew plant that lives in our greenhouse and eats tiny flies. So I finished the traits that I was taking in the other greenhouse, and now I am in our south greenhouse. Looks like a bunch of these plants are falling over, so I'm going to stake them up with some bamboo poles. So now they're standing up a little bit straighter. And here's another diversity panel that we have growing out in the greenhouse. We've got plants here from many different parts of silphium's native range that are wild. They haven't been selected here at all. So let's see. This one over here looks like it's from Iowa. And got one from Texas. So we're going to let these plants grow up in the greenhouse, and then we'll take trait measurements on them too. And we also collected some tissue for sequencing, marker sequencing. And here was where we're freeze drying that plant tissue from the diversity panel that we'll use for DNA extraction. Looks like things are still running. Now I'm going to take a lunch break. I like to go say hi to the cows sometimes, try and feed them some silphium, which I know that they really like. I'm going to do some spreadsheet organization now at my desk before I head back to the greenhouse. I'm back in the greenhouse this afternoon. I'm going to organize some of these pots and scan in the genotype of each of these plants into my tablet with my scanner and transplant the last few plants that are part of this experiment. I'm doing some transplanting from plastic pots to larger fabric ones. So I've left work and I'm stopping by the community garden now, which is collaboratively run by a group of TLI staff. This is a second sighting of a spider that's caught a syrphid fly in a bindweed flower. And here you can see some corn and squash coming up in the Four Sisters plot with seeds that were gifted by Indigenous seed keeper Taylor Keene. Taking care of the Four Sisters plot and the community garden is not part of my work responsibilities, but has helped me be a more careful and observant caretaker of plants. That is all I've got today. Thanks for tuning in to my day in the life at work of mid-June 2020, moment of pandemic and powerful uprisings against white supremacy and against capitalism. So I think in this moment I'm feeling the weight of the question of what are the new ways, what are the old ways of growing food that we need that don't treat land like a commodity, aren't modeled after plantation, and actually take care of people and of ecosystems. I will close this video with Abby's pet ducks.